The UPSC CSE arena is an ever-evolving space where staying up to date becomes as crucial as maintaining a thorough preparation. With strategic planning and holistic coverage for exam syllabus, Vision IS Delhi presents GS Foundation course. Avail analysis and integration of current affairs with GS syllabus. All India test series with an innovative assessment system, inculcating post-test analysis, discussion, all India ranking, and more. Explore the holistic learning modules intrinsically designed for the ideal aspirant.
Hello, good morning. So we are here to discuss Nobel Prizes. Every year in October you get this news, Nobel Prizes in few domains and 3 domains of science, which includes medicine, physics and chemistry. I'm here to discuss these 3 Nobel Prizes in today's class, okay? Without wasting any time, let's directly jump into the topic. The first set of Nobel Prizes, let's start with in the same chronology how it is given. So first, you have Nobel Prize in physiology or medicine, then you have for physics, for chemistry, and then of course other domains. So 2 scientists for the discovery of microRNA and its role in post-transcription gene regulation. Okay, this was an expected Nobel Prize. Like these 2 people were expecting Nobel Prize for quite some time now, because they discovered microRNA for quite some time, and it was a landmark discovery, and it was expected that somewhere down the line they will receive a Nobel Prize, and they finally did, right? Great scientists, great researchers. So let's try to understand what exactly this is. What are we trying to, what is the significance of this specific type of RNA molecule we are calling microRNA? And what do I mean by this word post-transcriptional gene regulation? Iska significance here. So let's try to understand these 2 keywords. And then of course we will try to understand that what is the significance of this discovery and what can be possible applications in different areas, okay? So to do that, I will go step by step. We will first brush up our basic concepts which we already have learnt in our biology classes. That think of the role played by these 2 very complex molecule, these 2 nucleic acid we call DNA and RNA, right? We have learnt about some aspects of, hopefully you have learnt about those things in more detail, right? But we will go step by step. So if I often start this topic in describing that, think of the biomolecules of your body. So your body is 70% water. Remove that 70%, you have many type of organic compounds, right? But all of those biomolecules, they can be mainly divided into 4 categories. The first category is called carbohydrates, which gives you immediate energy. The second category is called lipids, which includes fat, by the way, where energy is stored. At the same time, lipids also play a very important role in making cellular membrane, that lipids regulate what can enter inside cell, what cannot enter inside cell. But the third And one of the most complex biological molecule which is found in all living organism is proteins. And proteins do so many things. So while carbohydrate is the source of energy, immediate source of energy, or lipids are energy stored in the body or cellular membrane, proteins do so many things. You have eaten something bad, body produces specific enzymes to digest that. That is protein. You are working out, you have built your muscle. That muscle which you see, that is actually a type of very specific type of protein. You're feeling very happy, some hormones are released, that is a protein. You are infected by some pathogen, your body is producing antibody, that is a protein. So your body is this great protein making factory. And protein molecules are very complex. You cannot simply, there are protein molecules, forget about writing their name and structure on this board. I will not be able to do that if I start from there, that part of the wall, and go to there. They're so complex. So protein molecules are very complex. But the best part is that your body can make thousands of types of them. So our body literally is a protein making factory, right? But all of that happens because of the most important fourth biomolecule, which we call nucleic acid. And we have two types of nucleic acid depending upon their molecular structure. One of the nucleic acid is called DNA and another is called RNA. So we are going to talk about first DNA and RNA. We will try to understand about this word gene regulation or gene expression first. What is genetic expression? And then we will come to this topic of that what do I mean by the word post-transcriptional gene regulation with help of microRNA molecules. So let's go step by step. Let's start with this very basic thing which you already know of. You can think of DNA molecule, which is a fundamental molecule important for, so you can think of DNA as this, your blueprint, like whatever you are. Like we all look very different from each other. We all have different set of skills, different sets of aptitude. We learn those things in our life, but many of those things actually are simply because of our genetic footprint. So DNA molecule is what makes all of us unique. And we, by the way, share a significant part of DNA. What do I mean by the word sharing here? I will talk about that also. So we know a few things: development, functioning, growth, reproduction for all living organisms, and many viruses also. There are viruses which only have RNA. They're called RNA viruses. But then there are also, of course, many other viruses. So even viruses which are considered not living or also not non-living, but even many viruses are DNA viruses. So for all living organisms, including viruses, not all of them, but some of the viruses, DNA plays a role in development, functioning, growth, etc., etc. What this molecule is all about, we do not have to go into a lot of detail. For example, I have provided the information about that DNA is a molecule, and for every molecule we try to understand the molecular structure. That what is the molecular structure of a molecule? Carbon dioxide ka molecular structure is CO2, right? H2O, things like that. But DNA is a very complex molecule. But what that complexity is, that if you take one small fragment of DNA, I'm using this word rungs here. You can think of DNA as a ladder and think of that one stick, one ladder, and then what it will be made of. There will be a sugar molecule. We call that sugar deoxyribose, jahan se naam aata hai, right? There will be a phosphate group. So phosphorus is very important for DNA molecule. But I only want you to focus upon this last part, a nitrogenous base. That two DNA molecules are coiled together with help of these four nitrogenous base pairs, which means that nitrogen is essential to all four of them: adenine, thymine, cytosine, and guanine. And these base pairs occur in a, so now let's go to the structure part. DNA molecule, but a double helical structure with these base pairs. We call them ATCG, like adenine. So the best part is that adenine always goes with thymine and guanine always go to cytosine. And ये जो आपको बीच में अगर आपको pentagon दिख रहा है, that essentially is that sugar and phosphate group, which I can further zoom in this picture. So this is the actual structure of one rung of DNA. If I take that few parts here and zoom in into this, this is what I'm going to find. I'm going to find deoxyribose sugar. I'm going to find this connection with phosphorus. And I'm going to find these base pairs, but A always goes with C, A always goes with T, C always goes with G. So when I say that most of our DNA is more or less same, then essentially I'm talking about this base pairing sequence, that this base pairing sequence is more or less about 99.9% same for all of us. But that 0.1% difference, which also comes out to be about few million base pairs, that 0.1% difference is what makes all of us unique in different ways, like our eye color or our height or our hair color or our aptitude also. A lot of our aptitude is, some people have very good aptitude in music, or some people are really great at athletics. And it should be very clear that a lot of these things depend upon your hard work, practice, but a lot of that also depends upon your genetic footprint. So anyways, we know about this. So we immediately come to this part, that DNA carries our genetic information. DNA can replicate itself. When a cell divides, it makes a perfect copy of DNA most of the time. So every time many cells are dying and many cells are, your body every day is making millions and billions of cells, and many of them are dying also. Like our body has about 40 trillion cells, right? So DNA can replicate itself, but the part I want to highlight here is that one of the most important function of DNA is this part, protein synthesis. Inside your cell, if you zoom in inside your cell, then you will find that cell का खुद का अपना एक fortress है, जो cellular membrane है, which does not allow a lot of things to enter. But then when you go inside, you find that almost about at the center, you find this another structure which we call nucleus. And nucleus का fortress और strong है. So आप ऐसे समझ लो कि पूरा cell अगर एक city है, और city में बाकी बहुत सारे लोग हैं, तो ये जो part है ये basically castle है जहाँ पे एक king रहता है. And that king is a great dictator. Like a dictator, ये democratic king नहीं है, ये dictator है जो सब कुछ dictate करता है. Without this dictator, nothing happens here. So inside this nucleus, you have this molecule called DNA. So DNA is this entire instruction written in the nucleus. And one of that instruction, of course, and as we live, the most important instruction here is that protein synthesis, that your body can produce thousands of types of protein, and which protein needs to be made for what purpose, all of that information is stored in DNA. So not entire DNA is used simultaneously for protein, and this is where a part of DNA, a functional part of DNA, which we call gene, comes into place. So you can think of DNA as this very thick book, and you can think of gene as individual chapters. That, let's say when you study science and technology as a topic, there are individual or if you think of UPSC preparation as a thick book, and within that UPSC preparation, then there are individual chapters for each and every topic. So you can think of genes as those individual chapters, that these individual chapters are where the information about which protein needs to be made for what purpose, it's stored. So you can think of gene as the functional unit of your DNA. And when I use the word genetic expression, then this essentially it's saying that how a gene finally leads to production of a protein. So this word genetic expression is very simple, that the mechanism through which a particular gene leads to production of a protein. But this process also, this process where this information which is stored in one functional part of DNA, I'm calling it gene, helping in protein synthesis, that also happens into many stages. And those stages are very, very complex. And this is where the second nucleic acid molecule you have come across, that emerges. Few more things about gene. Instruction manual, lots of different sentences. Basic unit of inheritance, this is what you pass on to the next generation. specify physical and biological traits. Many genes code for specific proteins or segments of proteins. And we have about 20,000 protein coding genes, human beings. For different organisms, this can differ, but human beings have about 20,000 protein coding genes. But this process of genetic expression and final protein production happens into multiple stages. That when a protein needs to be made, the gene which is responsible for making that protein must be activated. So DNA molecule is this dictatorial regime which knows everything, it regulates everything, but it does not get out of the nucleus. It has many messengers and it does not send all the message to one messenger. इसको पता है कि किसको क्या मैसेज भेजना है. समझ लो, ये अगेन ये बिग बॉस है कि सब कुछ रेगुलेट करता है और इसको सबका पता है कि सब कहाँ पे क्या कर रहे हैं, क्या करना है. It knows everything, but it does not go and tell them. It sends its messengers. So this is where this first step in genetic expression which occurs, which we call transcription, that for a specific protein, a specific messenger RNA will be transcribed. In biology, we call this process transcription. And this messenger RNA now can go out of the nucleus and can go to the relevant organelles, and it can help in protein production. So when this part is called transcription, and finally, when this another organelle, ribosome, which makes a protein, we call this translation. So this entire complex process, without going into all of the technicality, what you need to know that protein production happens through transcription and translation. That you have a messenger RNA molecule, and then you have a finally protein, and that protein essentially has a very specific function depending upon for what purpose it has been used. RNA molecule is little bit different from DNA molecule. There are certain differences. That सबसे पहले तो single strand है, DNA is a double stranded molecule. RNA molecule also has bases, those nitrogenous bases. It has a phosphate group, it has nitrogenous bases, but it has uracil, right? So 3 bases are same, but one of the base here is uracil. And RNA molecules are of many types. This is where this Nobel Prize comes into place. Messenger RNA carries the genetic code from DNA to the ribosomes where proteins are synthesized. Ribosomal RNA forms the core of the ribosome structure, catalyzes protein synthesis. Transfer RNA brings the correct amino acid to the ribosome during protein synthesis. The RNA for which this Nobel Prize was given is this microRNA. Okay, so now let's come to this part. What is the significance of microRNA? microRNA का significance एक जो हम सबसे आराम से हम सब appreciate कर सकते हैं. But think about the fact that every cell in your body, or almost every cell in your body, has the same set of chromosomes, has the same 23 pairs of DNA, right? But nonetheless, a liver produces different protein, and a kidney produces different protein, and some other organ produces different protein, right? So everywhere you have same DNA, but the protein production is different depending upon for what purpose it is being used. A kidney will use it for some other purpose, a liver will use it for some other purpose. And this is where this microRNA molecule comes into place. That this microRNA is called microRNA because it literally is very small. That microRNA hardly will be like 20-22 nucleotides, not more than that. So it is microRNA. So how does microRNA, 22 nucleotides long? This microRNA is not coded for protein. That is messenger RNA, जो अभी हमने थोड़ी देर पहले डिस्कस किया था, for which Nobel Prize already has been given in past. For messenger RNA, ribosomal RNA, for all of these discoveries, Nobel Prizes have been given in the past. What is microRNA? This regulates the expression of other genes by binding to their messenger RNA and preventing them from being translated into protein. This is called post-transcriptional gene regulation. That some messenger RNA do not need, even if they are being transcribed, they should not lead to translation of a protein. This is the job of microRNA. That if a messenger RNA is being transcribed, but then this protein is not necessary in a specific organ. It is important in some other organ, but it is not necessary in this organ. And that part is done by microRNA. And that is remarkable. So this was a problem which was known to biologists for quite some time. They understood genetic expression. By the way, the first person who did that was a scientist of Indian origin. His name was Hargobind Khurana, and he received Nobel Prize in Medicine way back, I think in 1968. So this concept of genetic expression, he was the first person to determine that. He was the first person to know about the ribosomal or I think transfer RNA structure. So तब से Nobel Prizes बहुत सारे मिल चुके हैं इस पूरे डोमेन में. तो but एक जो problem जो open था वो ये था that how different organs regulate the protein production even though everybody has, all of the organs have same DNA. And this exact mechanism these two scientists were able to figure out, and that occurs with help of this microRNA molecule, that it regulates the expression of other genes. This in itself is not RNA which is coded for protein. तो बहुत clear होना चाहिए that microRNA is not coded for some protein production. MicroRNA is coded to bind to some messenger RNA, and then it degrades that messenger RNA. It stops that messenger RNA in making a protein, and that, by the way, is very essential in your body. And this is not just there for human beings, it is true for all living organisms, that all living organisms, even, so for example, these two scientists, they started their study on a very small worm, a 1 millimeter or 1 inch worm. And then they were able to understand that how same worm, but how different genes are being regulated. And then they, this became the base of their knowledge, and they applied that to other organisms, and they were able to figure out the structure of microRNA, the function of microRNA, and there were also more and more information came into light that how exactly this process happens, all the step by step how it happens. So it took many years for everything to be developed, but these two were the first person to give this term microRNA and talk about exact function of microRNA. So I will give you a few examples here. This again, this is not at all you need to remember. हमको बिल्कुल ही याद नहीं रखना है. Just to give you some examples. That ये जो हमने एक जो पहले मैंने एक स्टेटमेंट अभी बोला, that different cells perform different functions, and this is basically one of the most important reason they do that is because of microRNA molecules, even though they contain the same DNA. जैसे in pancreatic cell, microRNA molecules are named as they are discovered. So एक कोई भी microRNA molecule का नाम देखोगे तो एक ये लेटर आएगा, नंबर आएगा 375 जैसे for example. So this was this 375th microRNA molecule which was discovered. And then एक शुरू में भी एक आएगा script which will be basically in which organism it was found. So जैसे अगर यहाँ पे HS लिखा है, it means Homo sapiens, that this was found in Homo sapiens. कोई और species का कुछ और shortcut होगा. So that is the complete name of a microRNA molecule. Do not have to remember any of that. That should be very clear. This is only and only to give you some examples. Then pancreatic cell, this MIR375 regulates insulin production. MIR302 in an unborn baby regulates pluripotency of stem cells. And when we become adult, that MIR302 does not get produced in our body. It decreases significantly. And that pluripotency of stem cell, that almost comes to non-existent. So we know that in adults we have some multipotent stem cells, but we do not have pluripotent stem cells, and that is because that as we become adult, this microRNA is not produced. We know this also. Or this MIR146A regulates our immune system's response. So there are many examples of micro. Now we know hundreds of them, and we know for all of those hundreds of microRNA molecule exactly what purpose they are serving in human body, or even for other living organisms, right? So this is the significance of this discovery, and this was recognized by Nobel Committee, and these two scientists received Nobel Prize. So they started this study on a worm, this 1 inch worm, C. elegans, and they were able to discover a microRNA called Lin4. Do not worry about that. This is also not important. And this, of course, led to understanding the cell differentiation immune response. But why this is significant? Because now this discovery allows us to understand various types of diseases. That if microRNA regulation is not happening in the way it should in any living organism's body, then that will lead to various type of genetic disorders, various type of diseases, including some type of cancers. And that immediately also tells you that this discovery allows us to find better therapeutic medicines, that for if we know the root causes of these diseases, then we can also find solution to these diseases, which can tackle these diseases at its very core, rather than only treating the symptoms. So this is the significance of this discovery, that it allows, first of all, it allows us to understand this cell differentiation. It allows us to understand various other life processes which occurs in living organisms. It allows us to understand the root causes of many type of diseases. And then it allows us to make better therapeutic medicine to tackle those diseases. Right? So this is the role of microRNA molecule in this entire discussion about transcription and translation. So microRNA is also produced inside nucleus, but then it is carried to cytoplasm after that. And then there are many steps involved here. But finally, iska jo main purpose hai, that it degrades a specific messenger RNA molecule, that it shouldn't lead to a protein production. And in some cases, even if it is not able to completely stop the protein production, it definitely reduces how much protein is going to be produced inside the cell. So this is about the first Nobel Prize in medicine. If you have questions for any of the topic we have discussed, you can ask questions. This discovery helps in brain cells. I'm very sure that yes, it definitely can, because even brain cells are, we do know that neuron cells and brain cells have specific function. What type of proteins they are producing, without a doubt microRNA molecules are playing a role. And I'm very sure that we know about some microRNA molecules which play an important role in regulating production of proteins in brain cells, without a doubt. To what extent we know that part I cannot answer. But I'm very sure that you can find this with a simple Google search, that which microRNA molecules play a very important role in this post-transcriptional regulation of brain cells. That information should be easily available